Good morning, everyone. Thank you for joining our public call today on the Act for ALS and the ALS Strategic Priorities Community Updates. Please note that this public call is being recorded. The purpose of today's call is to provide a summarized update on the implementation of the 15 ALS priorities that were identified at the beginning of the year in the NIH LAS Strategic Plan. We will hear very briefly from representatives at NIH, the Foundation for NIH, the FDA, and the Critical Path Institute, along with pre-recorded updates from an expanded access program investigator and some of the nonprofit organizations with programs that align with the 15 shared ALS priorities. We will have two separate periods for questions from the audience, and you are more than welcome to enter those in the Q&A at any time. We are very interested in getting your comments about how we're doing in the implementation of these priorities. We have many folks on, so we might not be able to get to all of the questions, but please do feel free to reach out to us and we will follow up. I would like to introduce the Director of the National Institute of Neurological Diseases and Stroke, Dr. Walter Koroshetz, who will provide an update on the NINDS efforts as they relate to Act for ALS and the implementation of the ALS strategic priorities. Dr. Koroshetz.
Great, thanks so much. And can I have the first slide then? It's really exciting to be able to bring everybody together today. I went through the slides. It's just amazing how much good research and how much good advocacy efforts are going on in the ALS community. I think NINDS is certainly blessed with such strong partners, and we hope to be able to work with you on our shared agendas and to coordinate the effort to develop really effective treatments for ALS, both for those folks who unfortunately are suffering with the illness now, try and help them make their lives livable, more livable, and also to develop treatments for people who are at risk for ALS or are destined to develop ALS in the future. And in the best of all possible worlds, of course, what we'd like to do is to be able to diagnose the disease as soon as it shows any signs and then to stop its progression. So that's kind of the holy grail I think we're all moving towards. Each of the organizations is contributing in a major way. And I think that coming together will be stronger than we are working in our individual paths. As you know, we developed a strategic plan, we meaning you, and academic investigators, people with lived experience, and the ALS nonprofits. Next slide. So I'm pleased to show you on the next slide. No, not that one. One before that. The funding that has been moving towards ALS, and this is a pie chart. It's a little bit complicated. But there is the dark blue is the funds that NINDS releases for ALS research through a variety of different mechanisms, which we'll talk about in the future slides. Then there's the funding in that lightish blue that comes from the Office of the Director of NIH that's administered for the Act for ALS. So that's about 16% of the funds that go out. Then there's what we call the NIA funds administered by NINDS. And these are funds that are appropriated by Congress to the National Institute of Aging, but NINDS manages about 10% of their funds for dementia. And we fund research in frontotemporal dementia and the overlap with ALS. And then NIA in the gray also funds research in this space. And there are a number of other institutes that also have research programs related to ALS in the other colors. So the good news is we're on an upstream. We are, of course, worried about the budget for NIH. The president's budget, the Senate budget has us pretty much flat, which unfortunately, because our grants go out for 5 years and the costs go up in ensuing years, a flat budget really is a cut in the amount of money we have for new grants. And then there's also some question of whether NIH might receive a cut, which would be much more serious. But we are hopeful. We have grants that are out the door. We have really good investigators. We have great partners, and everyone is really devoted to making a difference for people with ALS. On the next slide, you can see kind of the major areas that came out of the strategic plan. And so certainly for NINDS, our strong suit is accelerated research on the biology behind ALS, trying to develop what we call targets from the biology that could potentially form the basis for therapy development. NINDS is not a pharmaceutical company, but we do the research that allows the pharmaceutical companies and the biotech companies to move from the discoveries at the bench to developing therapies that can then be tested in patients. We do have a number of programs where we are what we call de-risking that process, allowing biotechs and academic access to resources and funding for them to start off developing a treatment with the goal that if it's successful, that it would be picked up by a larger pharmaceutical company and brought into clinical trials. We also are very active in trying to understand the needs of the researchers to optimize the clinical research that's done ALS. I guess the best example would be in developing systems that would enable clinical trials to be successful, or developing systems that will allow us to get at really important biomarkers that can be used as measures when you go into a clinical trial to know if a therapy is really doing what you hope it's doing. And that's actually one of the biggest things that the industry is looking for from NINDS is this biomarkers that can help them in determining whether their therapies are actually hitting their targets. That's not something the industry has the ability to do because it takes a long time. There's no financing, financial incentive for that except to make the clinical trials better. But that's really a big part of what we think NINDS's role is in getting better treatments out. And then, of course, as I mentioned, we're the stewards of the taxpayers' funding for biomedical research, and people who are suffering with ALS are at the top of our priority list and trying to improve their quality of life. And finally, we're here together particularly in terms of this last bullet, which is to improve the collaborative partnerships and make the effort to make ALS a livable disease and to prevent ALS in the future, something that is going to be much more successful working together than working alone. And so we really value these partnerships and look forward to the discussions throughout the rest of the day. Next slide. So there are a couple of points in terms of the strategic plan that come out of those bigger bullets that you saw on the last slide. First of all, we've made a lot of progress in understanding the genetic causes, but the sporadic causes of ALS are really still a mystery. We need to learn from the genetic causes in the biology that leads to motor neuron disease from gene mutations to understand how it translates to the largest number of cases which don't have a genetic cause, but the underlying biology is likely quite similar at some level. This also is important. We're actually starting a program today, trying to understand what's occurring in the environment that could give rise to neurologic diseases, so-called the exposome. And I think that there's a lot of interest in, potential chemicals, certainly in the environment, infections in the environment that could be the cause of sporadic ALS that need to be pursued. And so that might be a good target for this new office that we've created at NINDS. We really want to understand the clinical heterogeneity and the molecular mechanisms that contribute to it. So ALS, unfortunately, has a fast progression, but there are some folks that have a slower progression. So to understand what mediates that slower progression could lead to potential therapies, for instance. And we want to harness emerging technologies to get at the answers that are necessary for the ALS community. Many people have worked in ALS for their careers, tried to answer questions, just can't get there, but the new technologies often offer the opportunity to get answers where that was not possible before. And there are also technologies, particularly in the brain-computer interface space, to allow much more higher quality of life in people with ALS. And we want to also take the opportunity to work with those folks who have a known genetic cause, but yet have no symptoms. Of course, the goal would be to prevent the disease from ever coming on. That's very hard to do in sporadic because you'd have to start with someone once they have a symptom. But in the pre-symptomatic case, one could potentially start treatments really, really early on. And this is something that I think is going to be tested very shortly in some of these genetic forms of ALS. And I'm so encouraged by talking to some of the doctors who tell me that some of these treatments for the genetic forms do seem to be working. People actually getting better, which is really quite remarkable in this small subgroup of patients with genetic forms being treated, say, with antisense oligonucleotides. We want to optimize the design and performance of clinical trials at all stages of the disease, including the pre-symptomatic gene carriers. And with the partnership of industry, we're focusing on the phase 2 trials to show proof of principle of effectiveness of a therapy. We're working fantastically with CPATH, the Critical Path Institute, works with the FDA to develop better outcome markers for phase 3 trials that could then come to the FDA for approval of therapies. I mentioned the importance of the biosamples, and there was a call to try to coordinate all the great work that's been done around the country and actually around the world in terms of bringing data and biosamples together, kind of one-shop stopping, not to actually put them in the same place, but develop a portal where someone could go to one place and see everything that there is, and then go out and get what's needed to answer the question. Certainly, we have valued tremendously from the input of people with lived experience in the development of the strategic plan, and we continue to bring that along in the current programs under Act for ALS. And again, looking for your input, how to do that better here. We're also working with academics and investigators, the FDA now much more closely, disease organizations like yourself, and under the Act for ALS Public Partnership, we have industry people who are also now committing with us to work on better treatments for ALS, which is I think a real bonus of the Act for ALS Public-Private Partnership. And finally, the National Academy of Science and Medicine is now doing multiple workshops, come up with recommendations on how we can improve the daily living of people with ALS and their caregivers, and identify interventions that promote maximal function and daily participation. So lots going on in the ALS space, in concert with the strategic recommendations that were made by by the working group of our council that worked over a year to develop those. There are a number of programs that NINDS has for researchers to come in, and some of them are listed here, and they range from transformative research projects to small business grants to develop therapies, training programs, programs particularly in the Alzheimer-related dementias that includes FTD ALS. This has really been a big boon to funding and also bringing people together. The frontotemporal dementia folks who are usually in memory disorders clinics now working with the investigators caring for people with ALS, because some of the genetic forms actually cause both diseases, and there is significant overlap there. The BRAIN Initiative is probably the most exciting and high impact program that NIH has ever done in the neuroscience space. It would take a while to explain it, but just to say that the tools and the data that's coming out of the BRAIN Initiative are being adopted by almost all the investigators looking at different diseases. So trying to understand how the cells in the brain are affected in disease is now really doable on a really gigantic scale, where you can look at single cells individually and say in the ALS space, the opportunity to look at motor neurons that are still healthy, motor neurons that are just on the verge of dying, and motor neurons that are in the process of becoming sick, in autopsy materials from people who've died with ALS. This is something that no one could have imagined could be done 5 years ago. And similarly, brain-computer interfaces now, which can code someone's thoughts about what they want to say into actual speech through a computer. So early days for some of these things, but there's no question that these are going to be very important for ALS research. We also have an intramural research program with investigators very interested in ALS and frontotemporal dementia. We have biorepositories where we hold biosamples of all different types. They're at the University of Indiana in a building inside of a building, and the inside building is at minus 80 degrees, and everything inside is robots run. You can't have people in there. It's way too cold. But those samples are open to researchers around the world. And similarly, we have clinical research going on to develop better biomarkers to understand phenotype, genotype studies. And the new research, which we'll talk about in a second, that's come out of the fantastic funding through the Act for ALS. And we certainly appreciate all the work that the nonprofits have done to make that a possibility. This is probably not news to anyone on the call, but just to mention that in trying to tie the genetic forms to the sporadic forms, the emphasis has shifted to a protein called TTP43, because that aggregates in both the genetic forms and the sporadic form. And there's evidence that it actually impairs the processing of messenger RNA, which is what is then going on to develop proteins. And these what we call cryptic exons could in fact be toxic to the motor neurons. And so this is an area of intense investigation now. It's not entirely clear how it's going to play out, whether it's cause or effect, but there's lots of interesting possibilities, including this one, which was reported out of the group in San Diego, showing that this abnormal cryptic exon in a protein called Stathmin actually prevents motor neurons from sending out their axons normally. And this can be corrected with an antisense oligonucleotide that prevents this abnormal splicing. So this is, again, as you see here, it's in the culture dish, but these are the kind of things that have to be pursued to the end to know if they're really going to translate into a robust treatment. But here you can see the kind of beginnings of how these things happen. We're also working with multiple groups here to develop better measures to chart the progression of ALS. Very important in developing, say, for instance, recommendations to the FDA about what would be a clinically meaningful difference of a therapy. You really want that to affect. You really want that to affect the things that make important, that are really important to people. Next slide. And then let's turn to the access to critical therapies for the ALS Act. And once again, thanks so much for your advocacy in moving this to fruition. So there are two parts to it. One are expanded access grants to enable persons who are not eligible for phase 3 clinical trials to take advantage of investigational drugs should they wish to do so. This is what's called expanded access. And the second part is a public-private partnership for rare neurodegenerative diseases to be set up between the NIH and the FDA, focus on advancing regulatory science and scientific research that will support and accelerate the development and review of drugs for ALS. So the funding here is generous, but it's very confined. It's it's very clear to us that the funds must go to expanded access first. And if funds are available, then they can be moved to the public-private partnership to support research on that would accelerate and the development and review of drugs for ALS. We have four expanded access research programs underway. One funded in 2022 and 3 funded in 2023. And you'll hear from Dr. Paganoni a little bit about the expanded access program in the ensuing time we have. Now, if we turn to the public-private partnership, we feel that a number of the strategic priorities can be addressed through a public-private partnership best, actually. And for instance, the strategic plan called for a network for ALS translational research, and we have been able to set up with these funds this ALS consortium to do just that. The strategic plan called for enhanced biosample and data infrastructure, and together with the Accelerated Medicine Partnership at the FNIH, we'll be able to do just that, which is to bring data, not just data that NINDS produces, but data that all the groups produce into a portal so that it can be seen in a very coordinated, comparable fashion, and biosamples can be accessed through a single portal, all the work that the different partners have done hopefully can be enhanced by this infrastructure. There was a real call to define natural history from people who are at risk of disease, so people don't have symptoms but have genetic risk, to discover the earliest manifestations of ALS. And importance here is that this gives us the opportunity to pursue how to improve the diagnosis of ALS so that people don't wait 6 to 9 to 12 months before they are introduced to the medications that have been shown to have benefit in ALS. And this is really important for research as well, because we want in any therapy to try to develop and test that therapy in the earliest stages of the disease. So trying to move to get an earlier diagnosis is very important for so many different reasons. We'll also be able to develop a platform for tools and protocols and resources that people can adopt. These will be vetted throughout our multiple investigators, our multiple groups, yourselves, and then made available. The platform will contain data, we hope, that much of which you have collected for clinical data, genomic data, imaging, biofluids, biospecimens, iPS cells, and autopsy tissue. We're interested in developing a pre-competitive collaboration. So this is not to be commercial, but it's to incentivize the commercial organizations, the biotechs, and the pharmaceutical companies to see that ALS is an area that is fruitful for therapy development, and the tools are available for them to be successful as they try to do so. And we want to use all our resources to provide, bring them together so they're not isolated and their value limited to the smaller number of people who see them, but to bring all these resources from clinical trials together so that everyone can benefit from the data that's been collected from patients. Next slide. So the consortium, we're trying to develop sites across the country, and we had really great applications, and we're basically limited by the funds we have, but we really wanted to have geographic diversity. So we have one site in the East Coast, one site in the West as the coordinating centers, and they have hubs that they will be working with. And I'm hoping that we can build this out over time so that the idea that a person with ALS who's interested in research, no matter where they live, can take part. So that's the goal. And we have different activities that are planned for these groups. Right now, it's the basics of collecting clinical data, biosamples. We'd like to improve our ability to collect really valuable or post-mortem tissues, but much of what goes on in the consortium is by a vote of the steering committee. So there is a group of industry, nonprofits, NINDS, and everybody's got one vote who joins. And so there'll be lots of things that come to that steering committee. And the question is going to be, what does the group feel is the most important thing? And so this is a fluid, dynamic situation, and is triggered heavily by the priorities of the people in the steering committee, both the industry, the nonprofits, and government. Should also mention, you'll hear from Colin in the time coming, that this public-private partnership is a component with the ALS consortium across the country of sites, then the Accelerated Medicine Partnership through the Foundation for NIH, which will bring the data in, and developing programs where industry funds will go towards particular research projects. And also the data will move to the Critical Path Institute, which we'll be talking to you later about their plans. And this link from early phase biomarkers all the way to potential phase 3 approval studies is something that can be coordinated through this public-private partnership in a way no one could have conceived of without it. So really a lot of excitement about this. I mentioned the Academy, the National Academies program going on now. Their report is to be released in June 2024. I'm sure many of you tuned in. The stories from people with ALS and their caregivers have been just so outstanding, and I think really make clear what the country needs to do to improve care for people with ALS as well. And I just want to end up by saying that here at NINDS, I feel so fortunate to be able to work with talented, dedicated people on this project, on ALS projects in general. And you'll be hearing from some of them in the time to come. But please know that our door is always open. Our ears are always willing to listen. We want to do the best thing with the taxpayers' money to make a difference for people with ALS, both now and in the future. So thank you very much. And really, you're going to really enjoy the rest of the sessions. And I certainly did reviewing the slides. So thank you, everyone. And now I'd like to introduce our colleague from the Foundation of NIH, Dr. Shubhan G. Lal. Shubhan G, over to you.
Thank you, Walter. Next slide, please. Good morning, everyone. I'm extremely pleased to be here and provide an overview of the Foundation for NIH or FNIH and the Accelerating Medicines Partnership for ALS and ALS. Next slide, please. All right, so established by the Congress in 1990, FNIH is an independent, not-for-profit organization that convenes public and private partnerships between NIH, academic institutions, private companies, and patient advocacy groups. It creates and manages partnerships that advance breakthrough discoveries and improve quality of people's lives. It has supported over 600 programs since its inception and currently has over 90 active programs within its portfolio. It has raised close to $1.5 billion in total investment to date. Next slide, please. The Accelerating Medicines Partnership Program, or AMP-ALS, or the AMP program, was set up in 2014 under the guidance of Dr. Francis Collins, the then director of NIH. At the center of the AMP program is a common goal of accelerating new and effective therapies to patients. It operates in the pre-competitive space and leverages resources of NIH and private partners to improve our understanding of disease pathways and transform current models for developing new treatments by identifying new targets, novel biomarkers, developing leading-edge tools and breakthrough technologies, assimilating large-scale data sets, and supporting open-source analytics, while generating consensus platforms and procedures. In the last 10 years, FNIH has led 10 AMP projects and raised close to $830 million in total investment, while working with 33 industry partners and 37 nonprofits across 16 NIH institutes. Next slide, please. As a component of ACT for ALS, public-private partnership, AMP-ALS seeks to expedite the development of effective new treatments for people living with ALS or at risk for developing ALS. AMP-ALS will focus its efforts on advancing the discovery, development, and validation of new biomarkers and clinical outcome measures to enable more efficient clinical trials and develop new therapeutic targets through aggregation and analysis of clinical data and biospecimens. This mission statement was developed by consulting people with lived experience of ALS and ALS advocacy groups. Next slide, please. AMP-ALS is currently in its design phase. The design phase was kicked off in June of 2023 and has active participation from public and private sector. Within the public sector, NIH, FDA, and critical path institutes have been actively engaged and have played a major role in forming this initiative. From the private sector, we have 9 pharma companies and 8 nonprofits that work in the ALS space participating in the design phase for this project. The steering committee in the design phase is led by Dan Doctorow, founder of Target ALS, Stephanie Fredette, who's the senior medical director at Biogen, and Amelie Gilberts, who is our moderator for today from NINDS. During the design phase, all the partners have worked through various working groups that were set up to develop a research plan. A PWLE working group was also specifically set up to ensure full participation of the PWLEs in the other working groups. We are currently in the process of wrapping up the design phase and begin active fundraising. with a potential launch at the end of Q1 2024. Next slide, please. So finally, there are a few key deliverables that will be an outcome of AMP ALS. First being the largest harmonized longitudinal ALS clinical data set that will comprise all stages of ALS, including pre-symptomatic and familial ALS. A multimodal molecular analysis of longitudinal biofluid samples, including postmortem tissue, whole genome, gene expression, targeted and untargeted proteomics data, new biofluid-based and digital biomarkers to assist in early diagnosis, monitor disease progression, as well as response to treatment, and new and novel patient-informed clinical outcome assessments. Next slide, please. Thank you, and looking forward to the Q&A. Back to you, Leo.
Thank you, Shubhangi. Next, we have Dr. Michelle Campbell from the Food and Drug Administration.
Thank you. Good morning, everyone. My name is Michelle Campbell, and I'm the Associate Director for Stakeholder Engagement and Clinical Outcomes in the Office of Neuroscience for the Center for Drug Evaluation and Research at FDA. Next slide. I will be spending the next few minutes providing the FDA update on the Act for ALS for the areas that FDA is required to do work under this Act. And so as Dr. Koroshetz said earlier, when he talked about what NIH is doing, part of Act for ALS has specific portions for both NIH and FDA. And I will be highlighting, or I should say our next speaker, Dr. Colin Havinga, will be talking about the public-private partnership, but I'll be focusing on our sections 4 and 5, which cover our action plan and our rare neurodegenerative disease grant program. Next slide. So we published last June of 2022 a 5-year action plan to foster drug development and facilitate access to investigational drugs for ALS and other rare neurodegenerative diseases. And in this plan, we laid out systematic goals of short, mid, and long-term goals to achieve various tasks in order to improve and foster drug development. We did focus on ALS, but this also does cover all other rare neurodegenerative diseases. We additionally have awarded grants and contracts both to public and private entities to cover costs in research and development of interventions intended to prevent, diagnose, mitigate, treat, and cure ALS and other rare neurodegenerative diseases for both adults and children. Next slide. So for our rare neurodegenerative program, during our first year of Act for ALS funding, we stood up our own task force internally and did a gap analysis of all of our medical product centers, as well as our National Center for Toxicology. And this effort is being funded and being housed out of our Office of Orphan Disease Products. And so we identified at that time 5 opportunities for investment, looking at comparability and equivalency studies for the use of clinical outcome assessments through decentralized trial aspects, natural history studies, the enhancement of devices with brain-computer interface, looking at FDA cleared or approved in vitro diagnostics, and also looking at basic and translational research related to the pathophysiology of rare neurodegenerative diseases and what is needed for the development for drug discovery. Next slide. So during our first year, we funded 3 grants and 2 contracts. And as you can see in the first 3 grants that went out, we did fund a natural history study looking at ALS, and it is both retro and prospective study. We additionally have funded efforts in ataxia telangiectasia, as well as myotonic dystrophy type 1. We also have funded 2 contracts. One that did a landscape analysis on BCI focusing on eventually if developing a patient preference study for use in ALS patients and what would be needed to begin that work, as well as an equivalency or comparability study looking at the ALS FRS and its potential remote use and standardization of remote use for clinical trials. Next slide. So for our this fiscal year that just ended at the end of September, we continue to support the prior works of the prior first year projects and provide additional funding to the natural history study. We also supported an additional clinical trial work for another rare neurodegenerative disease. And we also looked at 2 new grant opportunities, and we did have an RFA out for comments. We went through the comments and we really saw that the main interest that we heard from you, the public, was an improvement in the understanding of pathophysiology and the advancement of biomarker development from that RFA. Next slide. So we put out 2 funding opportunities this past spring. One was specifically on natural history and biomarker studies for rare neurodegenerative diseases. And there was a second one for looking at the systematic review of clinical outcome assessments for the communication of BCIs in ALS. Next slide. And so from those funding announcements that came out, we did fund 5 new awards this year. And you can see on this slide that a majority of them did go to ALS and ALS efforts. We have some work looking at BCI, additionally biomarker study work, as well as some work in the familiar ALS space, and additional work looking at biomarkers in myotonic dystrophy and Niemann-Pick type C. So again, we are continuing to look at efforts to fund rare neurodegenerative diseases, including ALS, and we're always trying to reflect where is the area of greatest need in order to fund these opportunities. So with that, next slide, I will turn it over to Dr. Colin Huvinga from the Critical Path Institute.
Thank you, everyone. It's nice to see some familiar names in the attendees list and also some new ones. So I'm hoping to update you a little bit on Critical Path, but also Critical Path for Rare Neurodegenerative Diseases, which is colloquially known as CPRND. Next slide. Just to give a quick snapshot of what Critical Path is that we were an organization that was established to work with stakeholders to create solutions to barriers in drug development. So we don't necessarily drive the agenda per se. What we do is we collect a lot of information from stakeholders, including folks like yourselves, and we synthesize those into a direction. And those directions basically, what's the problem with the rare disease? What solutions need to be built to make this more successful as a drug development platform? And then we work with those to address those areas. If you look at sort of our core competencies, obviously there's a diverse set of skill sets within Critical Path. Obviously, we have a backbone in regulatory science. It helps us understand what questions we need to frame into a context that may be suitable for a drug company or investigator to get a drug approved. Data management and science, and data in a way that's usable for various types of researchers, including for drug approval purposes. Biomarkers, not in the discovery piece, that's not our wheelhouse. That's NIH and FNH's world. But what we do is we look at biomarkers in a specific lens called the context of use to decide, is there enough data to justify this biomarker for a particular strategy? And then we work with regulators to get those biomarkers endorsed. Modeling and simulation, I think one of the challenges everybody on this call can appreciate is that when you enroll in a clinical trial, obviously one want to figure out a trial that's the shortest as possible as it can be to be scientifically sound, as well as minimize the exposure to control groups or placebos. And so one of the things we do is we create simulation tools that help address those elements. And then, of course, as Dr. Koroshetz mentioned a bit earlier, is that we also look at new novel endpoints that are meaningful and concepts meaningful to the persons with lived experience community, so that we're trying to understand the things that are being measured that are meaningful to impacted individuals. Next slide. Just give you a quick lens shot of what this looks like. I mean, obviously we talked a little about data and other presentations. It's sort of a core element of how we function. We work through ingesting of data. Then we can produce any of those specific tools with working groups aligned to that end. And so, we typically involve advocacy groups. We involve sponsors. We involve regulatory agencies. We involve scientists. And et cetera, all to create something that will meet regulatory rigor so it can be used to accelerate clinical drug development. Our hope here is to expedite the way in which clinical trials are accomplished, but also making them scientifically sound. And we're going to do that in a broader based community approach. Next slide. I want to emphasize something. This was nice to have a backbone conversation, that sort of the trail end conversation. We heard sort of all these different efforts going on. One of the things I will reassure the community is we talk a lot. So, obviously, each of our wheelhouse of what we're going to focus on may be slightly different. But if you look at each of the stakeholders that's presented on this call, we plan, we connect, we synergize, and we basically try to fill knowledge gaps. Sorry, I had to turn my camera. We try to fill knowledge gaps, together in meaningful ways, because nobody wants us reproducing things that have already been reproduced. We don't want to be tripping over each other, duplicating efforts. We want to make this a very communicated, strategic plan. And so as you look at that sort of circle, we see a lot of, activity going on, but it's all in lockstep to try to figure out the most efficient way to get to that cure that Walter noticed. As you notice, persons with lived experience are central to this. We have regular open forum discussions with members from NIH, FDA, and Critical Path all come together and FNH come together to discuss, what are people's concerns, update them so that they know what's going on. Because one of the things that I talk about and when we're in these meetings is that we're kind of your employees, right? I mean, you're taxpayers. And so we want to make sure that you understand and we get feedback knowing that we're in the direction that you need us to. Next slide. I'm going to go quick over this. I mean, I think part of this was an update. We did a ton of landscaping in conjunction with FINH and NIH in the last year. We spoke with FDA to understand their challenges in drug development. We did extensive amount of data inventory under finding like where is ALS at for drug development. And so given that, we came up very much in line with FNH, NIH, and FDA focus areas for Critical Path Institute. Next slide. Obviously, we're going to be focusing on drug development tools. We want to de-risk and attract sponsors to this space, but also expedite the way in which clinical trials are done through basically things like, biomarker qualifications, looking at standardizing clinical data so it can be used for multiple purposes in conjunction with AMP, ALS. Digital health technologies, obviously, that's a big area. We heard that a lot from the need to decentralize trials. We've launched a large initiative with the support of FDA around clinical outcome assessments, looking at with the ultimate hope of creating those simulation tools and advancing regulatory science in this space. If you look at our focus areas very quickly, because I want to give us some time for Q&A, we will be working with the person with lived experience in having their involvement in all the working groups going forward. And so we want to have that reality check and guiding star as we do things. Working at federating, our strength is in clinical data. I mean, obviously, CPATH has worked in federating data with multiple types. Obviously, our experience is a huge, landscape with data that we can bring, but also the strengths and the partnerships that you heard others mention is that the omics and the clinical data can be used, much more readily because of these conversations that we're having. Access to regulatory landscape, as we mentioned, we're really going to be trying to formulate tools and identify where there might be gaps where, NIH or AMP ALS can support additional funding. And then really come up with standards. Every time I'm on a call, I think with people, I keep thinking about the foundation needs to be laid a little bit more tightly on this, everything from methods and other things that we haven't had a real standardized ways to do it. And so we'll be working with investigators, academics, sponsors, et cetera, to do that as well. Next slide. I think that's it for Q&A. Hopefully we have some time for that and some really good questions I see in the chat. Thank you.
Thank you, Colin, and thank you all the speakers for these really excellent, informative presentations. We are running a little bit behind time, so we've been sort of working in the background. We collected all the questions, and what we're trying to do is to answer the questions in the Q&A box. And we also may have a separate session where we sort of just all get together to talk about these questions and give you answers. But right now, I think it's the best because we don't want to shortcut the presentations by both Sabrina Paganoni about the expanded access study that is currently ongoing, and then all the excellent videos and talks we received from our partners. So maybe let's move ahead in the agenda. And at the very end, there is still a Q&A session, so maybe we'll have a little bit more time. But otherwise, thank you for all the questions. We'll find a way to provide responses, and we'll be back in touch. But let's move over to the next presentation. Ilio, if you can get to Sabrina's presentation.
Thank you, Dr. Gubitz.
Hello, this is Sabrina Paganoni from the Healy and AMG Center for ALS. It's an honor to present an update on the Trehalose EAP. This EAP is up and running. Today, several people living with ALS are receiving Trehalose across the country thanks to this EAP. I work as principal investigator on this study with my colleagues Dr. Babu and Dr. Berry. This was the first EAP funded by the NIH under the Act for ALS. This EAP is happening in parallel to the efficacy trial of Trehalose. This means that the efficacy of Trehalose is being tested as we speak in Regimen E of the Healy ALS platform trial. We expect efficacy results from this trial over the next few months. EAP participants are not eligible for clinical trials. So the group of participants in this Trehalose EAP is different compared to the participants in the efficacy trial. Administration of Trehalose requires weekly intravenous infusions. This is also the first time that a large multicenter EAP for ALS includes weekly infusions. We're doing them at home to make this feasible and convenient for people to participate. The overarching study goal is to provide access to Trehalose and to collect research data. We plan on using the research data from the EAP to supplement findings from the efficacy trial. Specifically, research objectives include data on the safety of Trehalose in a broad population that is not eligible for clinical trials. And we're also going to study the biological impact of Trehalose on ALS by measuring neurofilament levels. And we're going to collect data on the clinical benefit of Trehalose. Target enrollment is 70 participants at 25 sites. I am pleased to share that in our first year, we met all NIH milestones ahead of time. In this first year, we developed the Clinical and Data Coordination Center and site network that are necessary to run this EAP. This EAP is run with the same rigor as a clinical trial, so that research data from the EAP can be used to advance the development of new treatments for ALS. We're going to supplement the data collected in the efficacy trial with data from this EAP. And this approach implies that we first had to set up rigorous central processes, including site selection and training, vendor contracts, central services to oversee the study, such as the single IRB, the central lab, etc. All central processes have been set up successfully and implemented, and the study open for enrollment as planned. So far, we have included 46 participants at 18 sites. This is a snapshot of the central infrastructure required for this EAP. You can see a list of the multiple stakeholders who have been working collaboratively and have contributed to the study, such as the coordination center, the single IRB, the vendors, and the study sites. I would also like to highlight the critical importance of the patient community who has been providing us with advice via Patient Advisory Committee. And I would like to thank the NIH, who has been meeting with us regularly and participates in the steering committee. And I would also like to acknowledge Silos Therapeutics, the drug manufacturer. This program builds on a few years of experience. Our vision is to make EAPs a reality for people living with ALS worldwide. Our team at the Healy Center began offering EAPs in 2018. This program was small and it was available at our center only. In 2021, thanks to the advocacy and support from the ALS patient community, we were able to expand our EAPs to a total of 9 sites across the US. These EAPs were funded by generous donations from patients and foundations and were a stepping stone towards the current EAP. The Act for ALS and the grant we received from the NIH in 2022 was the catalyst that allowed us to bring EAPs to a new level. This EAP is larger than prior EAPs. It is more complex. It also has important research goals and comprehensive scientific data capture. We believe that the Trialose EAP will serve as the blueprint for the design of scientifically relevant and patient-centric EAPs. Thanks to NIH funding, we're now able to offer EAP options at several sites. We plan on completing site activation and participant enrollment for this Trialose EAP over the next few weeks. This is a shift in paradigm because for the first time, EAPs are available across the country. To conclude, I would like to thank all study participants and all stakeholders who are supporting this EAP and are partnering with us to create new models of research access and participation in ALS. Thank you.
Thank you again to Dr. Sabrina Paganoni for providing that update. Now, just to recap, the 15 ALS strategic priorities were identified by the broad ALS community of researchers, advocates, the medical community, and people whose lives are impacted. As you know, many nonprofit organizations have ongoing projects that continue to align with these priorities, and there have also been new programs launched since those priorities were identified. What follows are some updates from some of those organizations that have programs with goals that align with the 15 priorities.
Hello, I'm Fernando Vieira. I serve as the CEO and the Chief Science Officer at the ALS Therapy Development Institute, and I will be updating today on ALS TDI's progress in 2023 against the objectives laid out by the NIH ALS Strategic Planning Working Group earlier this year. There were 5 key objectives laid out by the working group. ALS TDI has made substantial progress against 4 of those, and those will be my focus. First, accelerating ALS research to better understand the biology of the disease with an emphasis on unlocking sporadic ALS. ALS TDI operates the ARC study, the ALS Research Collaborative Study. ARC is the first and longest running ALS natural history study, which also integrates translational research using multiple omics technologies. The ARC study allows all people with ALS to enroll, and it is completely remote. Approximately 75% of participants in this study have sporadic ALS, with 25% being focused on genetic mutations, people living with dominantly inherited mutations, which are known to enhance risk of the disease. Using this study, we have the opportunity to compare and contrast various types of familial ALS with sporadic ALS, looking for common disease pathways and common therapeutic targets, and also distinctive therapeutic targets as well. These studies inform the drug discovery efforts that happen here at ALS TDI. We are working hard to understand the molecular mechanisms which underlie clinical heterogeneity. The ALS Research Collaborative includes digital outcome measures that allow for robust clinical phenotyping, where we can characterize types of onset, bulbar versus limb versus respiratory versus axial, and also allow us to relate those types of data to biological samples that have been collected from many of the same people with ALS. So we are collecting blood samples, and we are collecting samples which can be used to generate induced pluripotent stem cells from a very heterogeneous ALS patient participant cohort. This allows us to compare fast versus slow disease, bulbar versus limb onset disease, etc. We have been employing in 2023 robust proteomics and transcriptomics studies looking at more than 250 samples of each type from people with ALS, longitudinal and single time point, to better understand disease states and to reveal biomarkers that might predict each of these disease states. Also, a focus of accelerating research, we've harnessed emerging technologies to uncover new mechanisms. Like I said, under the ARC study, we're using genomics, transcriptomics, proteomics, and digital outcome measures. However, also through the Augie's Quest Translational Research Center housed here at ALS TDI, we have robust cell biology capabilities with a clear focus on using human-induced pluripotent stem cells from people living with ALS. We use those reagents to set up automated cell biology screens and high-content phenotype discovery. And we have recently implemented a new effort to deploy CRISPR technology to reveal genetic targets that might be disease modifiers, accelerating or slowing ALS disease progression at a cell biology level. We're also translating fundamental research into potential ALS therapies, as is an objective by the working group. One key approach was to establish a network of ALS centers of excellence to support ALS translational research. We submitted a collaborative proposal to an NIH funding mechanism with Answer ALS, Columbia, Johns Hopkins, Mayo Clinic, Emory, and New York Genome Center. This proposal was not funded. However, we at ALS TDI are now in discussions with funded organizations, Massachusetts General Hospital and Barrow Institute, to support the new all ALS natural history study efforts that will emerge in coming years and will be complementary to the ALS Research Collaborative efforts. We've worked hard to enhance and expand ALS biosample and data infrastructure. For our ARC study, our IRB has approved an expansion of the study to 2,000 people from our existing 1,000 participant cohort. We added electronic health records to the data types that we collect, and we created and launched a cloud-based data sharing platform that make these data available at no cost to academic and nonprofit organizations. We also have been sharing cell lines that we've collected through the ARC study with industry and academic researchers. We make our own internal assay systems and our expertise, our staff, available for screening therapeutic ideas from collaborators from academia and industry. And we have a robust preclinical pharmacology team, which allows for lead optimization and lead discovery in animal models, looking in mice and in zebrafish. We have dozens of active collaborations. Finally, we've made all of our ARC data available under the auspices of our ARC Data Commons, a cloud-based data sharing platform which can be used for target discovery and biomarker discovery purposes. We've worked to optimize ALS clinical research by now actively recruiting asymptomatic mutation carriers into our study, while also enrolling participants no matter how far progressed in their disease they are. We do enroll participants in the ARC study who are in later stage ALS. We believe it's important to understand both extreme states and every state in between. And as I mentioned before, we did establish our own multimodal data platform to share our clinical data, but we are working with the AMP-ALS enterprise to make our data sets available to a larger repository to serve the purposes of the ALS research community. We are working to better understand our own data set and understand gaps related to diversity, equity, and inclusion. In evaluating our own data sets, we do see that there are shortcomings, and we need to enhance the diversity of our participation by accessing underrepresented communities in the United States and expanding our program to enroll more folks globally. All of our progress has been made possible by our ALS patient community and who have enrolled in our studies and have supported our work. Thank you.
Good morning. This is Amy Easton, Senior Director of Scientific Programs for Target ALS. It is my pleasure to share with you today the progress we have made on the NINDS ALS strategic goals, a reflection of our deep commitment to defeating ALS. Over the past year, Target ALS has taken a dual approach to accelerating research on the disease biology of sporadic and familial ALS. First, 7 basic biology consortia, 6 newly independent academic investigators, and 7 Springboard Fellows received grants to study fundamental biology of ALS. The research from this talented group of scientists tackles a wide range of new concepts in disease biology using cutting-edge technology and innovative science. Second, Target ALS has invested in expanding our post-mortem tissue core resources. Our biobank contains samples from 190 patients with sporadic ALS, 63 mutation carriers, FTD cases, and healthy controls. These samples are distributed regularly to researchers in academia and industry and have been key to advancing our understanding of the disease. In addition, Target ALS facilitates research by sponsoring the generation of whole genome sequencing, bulk RNA sequencing, and now spatial transcriptomics and phospho-TDP43 staining. All of these data will be made available via our new data portal and analysis platform in 2024. These data will help shed light into cellular and spatial changes in brain pathology that may explain motor neuron susceptibility and differences in clinical phenotypes across patients. In addition to our efforts supporting research on the basic biology of ALS, we have taken a similar dual approach to optimizing clinical research. First, we have funded 6 biomarker consortia in 2022, 6 industry-led consortia in 2023, and we have 2 ongoing calls directed towards clinicians in the early stages of their career, both in their neuromuscular fellowship as well as in their first years of their clinical appointment. All of these recipients will conduct research to have impact on the development of novel diagnostic disease biomarkers, clinical trial design improvements, and ultimately the development of therapeutics for sporadic and familial ALS. Second, Target ALS is rapidly expanding our longitudinal biofluid and natural history study to follow disease progression in at least 800 ALS patients and 200 healthy controls. We have doubled the number of Target ALS participating sites to include diverse patient populations around the globe, and we have partnered with NIH to establish a nationwide network of clinics, allowing for broader access to both pre-symptomatic mutation carriers and symptomatic patients. With this effort, we will be generating the most comprehensive patient data set, including clinical, demographic, functional data, whole genome sequencing, biofluid multiomics, and neurofilament light. But the biofluids will be made freely available upon request in 2024. The combination of biofluids and data sets will fuel identification of novel biomarker and genetic or environmental factors influencing disease risk and progression. Since the founding of Target ALS, Dan Doctoroff has made it his mission to break down barriers to research and remove silos that can prevent progress in developing treatments for ALS. Consistent with this philosophy, more than 59 multidisciplinary consortia have been funded by Target ALS. These consortia have brought together academics and industry professionals, as well as technical experts from all areas of research. We are thrilled to share that these efforts are paying off. 60% of our consortia have supported pharma and biotech drug discovery programs, 7 programs have entered clinical trials, and 5 biotech companies have been launched. Collaboration amongst talented scientists has truly led to translation of fundamental research into potential ALS therapies. We are hopeful that our ongoing research will deliver the next wave of truly life-changing therapies. We sincerely thank all of the scientists, patients, and donors who have made this research possible. Thank you for your attention.
We appreciate the opportunity to provide the community with an update on our local efforts to support the NIH ALS strategic priorities. I'm Lauren Webb, Chief Advocacy and Outreach Officer for the Les Turner ALS Foundation. We are committed to providing the most comprehensive care and support to people living with ALS and their families so they can confidently navigate the disease and advance the research and prevention, treatment, and cure of ALS. The mission of the Les Turner ALS Center at Northwestern Medicine is to strive for a future without ALS by accelerating leading-edge research while providing life-enhancing treatment to people living with ALS. One of the top priorities identified by the committee was the need to better understand the biology of motor neuron disease. We fund basic science research grants at the Les Turner Center at Northwestern Medicine. The researchers at the center are collaborating with the NIH Center of Excellence programs at Feinberg School of Medicine to ensure that we are prepared to submit a proposal to become an ALS Center of Excellence when funding becomes available. We fund multidisciplinary clinic at Northwestern. It's one of the largest multidisciplinary clinics in the area with the highest number of neurologists and a dedicated pulmonologist. The clinical research program encompasses a broad range of work, including biomarker studies, open-label therapeutic studies, natural history studies, and clinical trials. They also participate in expanded access programs. The center was recently invited to participate in the All ALS Consortium. Being part of a large study will improve our understanding of clinical measures and biofluids from people living with ALS and individuals at risk of developing ALS. To optimize clinical research, the Foundation financially supports the ALS research program at Northwestern Medicine to help with staffing and data collection. The Les Turner ALS Support Services team plays a vital role in following the diagnosis, as they often become advocates for the person living with ALS and family, helping them to process the disease, clarifying information conveyed by the physician, and providing hope and support. We also serve on a number of committees to improve clinical research design, retention, and recruitment. Our team also encourages people to join the National ALS Registry, complete risk factor surveys, and donate specimens to the National ALS Registry Biorepository. We provide a personalized approach, preparing people living with ALS to navigate their difficult journey and listening to addressing their needs that they have right now and in the future. In June 2023, we launched a newly diagnosed series based upon feedback from the community. We are currently evaluating that program and will launch another group next year. In July, we started a monthly drop-in session where people we serve can receive guidance and help, connecting them with Shirley Ryan Abilities Lab's Assistive Technology Program, Team Gleason, and Bridging Voice. We also have implemented new processes to improve our loan closet for assistive technology. Using language informed by health literacy best practices, my ALS decision tool is designed to impart accessible information about ALS treatment options and to encourage conversations and facilitate careful planning of treatment decisions. Each My ALS decision tool was developed in collaboration with people living with ALS, caregivers, advocates, researchers, and clinicians. On May 1st, we launched My ALS Decision Tool on genetic testing, which is the first tool of its kind in the US that focuses on genetic testing for anyone with ALS. Clinical guidelines have changed, and we wanted to explain ALS genetic testing using health literacy best practices. After using the tool, people will have a better understanding of the benefits of genetic testing and the downsides as well. Users will be better equipped to make informed decisions about whether to pursue testing in partnership with their ALS care team. Since the publication of strategic priorities, we have hosted webinars on cognitive function, exercise, invasive ventilation. Please join us in December with Dr. Melinda Cavanaugh to learn about her research and findings to develop supportive interventions for the whole family. We also have a number of different guides developed with health literacy best practices for you to check out. In 2023 and 2024, we will continue to advocate for the ALS Better Care Act to increase Medicare reimbursement for multidisciplinary clinics across the country, and we hope that this will improve care provided to people living with ALS. We also plan on launching a genetic testing decision tool for people at risk of developing ALS. We also plan on working with NILS and ALS research ambassadors to design a research tool guide and clinical research decision tool. We hope that this will help people make decisions about clinical research. And lastly, we plan on enhancing supportive services in partnership with people living with ALS. We are currently surveying those we serve, and we'll have more information in the coming months. We continue to look forward to future collaborations and work together to find effective treatments and ultimately a cure for ALS. Thank you.
Hello and welcome to this presentation on the International Alliance of ALS M&D Associations. My name is Kathy Cummings, and I'm the Executive Director of the Alliance. We are a UK registered charity, and I'm speaking to you from my home in Canada, where I live and work, because we are a completely virtual organization. Let me tell you a little bit about the International Alliance. The International Alliance is an organization of organizations around the world. We have about 60 members in 40 different countries, and all of our members are ALS or M&D associations or societies that have a shared vision with us of a world free of ALS M&D. We enable global collaboration among Alliance members, people living with ALS and their caregivers, and internal and external partners through our councils, our events, our forums, and more. And we share knowledge, create resources, and advance research and care. Like the strategic planning working group that we're all presenting to today, all of our work is informed by people living with ALS M&D and their caregivers. We work with our members and partners towards more effective diagnoses, prevention, treatment, and ultimately a world free of ALS M&D. Let me also talk about what we don't do. We don't fund research. We are more of a curator or a conduit to an ecosystem of global key opinion leaders who work together towards our shared goals that we're aligned on. I want to talk to you a little bit about our advisory councils because this is how we do most of our work at the Alliance. We are a very small resource organization. I'm actually the only employee of the Alliance, and we do have a few contractors that support the work that I do, either on a part-time or a full-time basis, depending on their capacity. But we do most of our work through what I term our army of volunteers. And the majority of our volunteers, in addition to our governance structure, come through our advisory councils. We have 3 advisory councils. One is a PALS and CALS advisory council, one is an innovation and technology advisory council, and our third one is the scientific advisory council. So our PALS and CALS Advisory Council is a group of 10 to 15 people living with ALS, MND, and they have a number of projects, but their primary role is to be the keeper of the fundamental rights of people living with ALS, MND, and the fundamental rights of caregivers. These rights statements are available on our website, and you're welcome to check them out. Our PALS and CALS Advisory Council reviews them every year, and there are a number of satellite groups that come out of the work of the fundamental rights. And I won't go into the details of all of them because we only have 3 to 5 minutes today to talk about how our strategic priorities align. Our other advisory council is the Innovation and Technology Advisory Council. Again, about 10 people, including care directors from around the world, speech language pathologists, industry partners from tech companies, and people living with ALS. The biggest project that they're working on right now is a voice preservation project, and I'll talk about that a little more later. And they're also working on how AI can assist in many different areas, including care and research. Our Scientific Advisory Council is also around 10 people. It's clinicians, research directors, bench researchers, and people with lived experience. There are a number of satellite groups that we're in the process of setting up for 2024 to do some project work, and those are listed on the slide around genetics, clinical trials, clinical management, knowledge translation, and of course our Global Research Directors group as well. I want to pause here and just say that when we were asked about our priorities, meshing with the strategic priorities that were outlined for this group, we really could talk to all of them today, but I'm going to focus on 3. The 3 I'm going to focus on are collaboration and optimizing research and optimizing quality of life. So in terms of collaborations and partnerships, I moved this one up. It was the last one in the slide deck for the listed priorities, but I moved it up because I will cheesily say that it was last in your chart, but it's number 1 in our hearts. This is the raison d'etre for the Alliance. This is the whole reason we exist, is to enable collaborations and partnerships at a global level. All our councils and committees and our board of directors is made up of worldwide participants. We have people from every continent, every type of organization, from completely volunteer-led to multi-million-dollar operations. Every program we do, whether it's our Alliance meeting or our Allied Professionals Forum, roundtables, webinars, they always include people from various stakeholder communities, various geographies, and underrepresented communities, so that we make sure we are being inclusive of the whole world. We also work with other diseases, global organizations like the United Nations and the WHO, and with regional networks like UNELA and UPALS. We work with industry partners. Every single thing we do is an example of collaboration. We really do emphasize this in everything we do. I also want to talk a little bit about optimizing ALS clinical research. Again, I'm not going to talk about every project we're working on, but I do want to talk about a couple of key ones that I think are important in this particular space. We have a number of networks that we're supporting through secretariat work, ELATAM, which is in Latin America, and the ALS Africa Network, which are both emerging networks similar to NILS, NCALS, and PACTALS. We, of course, work with NILS, NCALS, and PACTALS as well, but just in a collaborative way. We don't support those particular networks in the same way that we do the new ones. We're also having a project on outcome measures and improving regulatory pathways. And we are also working on a project through our Innovation and Technology Advisory Council to bring voice preservation to underserved communities. And we want voice preservation to be available in the language of the individual's choice on a device that they already have and with no cost attached to it. A couple of other projects that we're working on are around being able to satisfy people's needs to participate in arts, music, sports, and culture. And a project we just recently completed was to be proactively prepared for emergencies, whether they're conflict or climate change driven. And again, the outcomes of this are available on our website. Thank you for your attention today. I do want to mention that if you have any questions, you can reach out to me at any time. And keep in mind one message from today, that the International Alliance is a unique enabler of global collaboration. Thank you.
My name is Emily Johnson. I am the Communications and Operations Associate at IMALS. Today, I will be providing you with a quick overview of IMALS, as well as how IMALS's work aligns with the NINDS ALS Strategic Plan. IMALS is community-led. Our volunteers create community teams around affinity groups and critical components of the ALS landscape. These teams meet weekly and are organized by volunteers. We also know that living with and being impacted by ALS is devastating and depletes resources. To retain our volunteers and ensure that they are the best advocates they can be, we needed to create programs and initiatives that provided support and directed them to resources. By organizing with the community, we ensure that the problems being addressed and the solutions being proposed reflect the reality of people living with ALS. IMALS's organizing efforts are led by the ALS community. There are 10 community-led teams that create their own agendas and action items. IMALS helps facilitate their work by creating advocacy tools and templates. In addition, IMALS advises on strategy and helps advocates become more effective and efficient. To support the ALS community, IMALS created the ALS support team, the Peer Support Initiative, and support groups. The ALS support team is comprised of 4 IMALS staff members who help people do the following: process the emotional impact of ALS, learn more about clinical trials and accessing them, and finding assistance with finances, insurance, and legal concerns. IMALS's Peer Support Initiative connects people impacted by ALS with each other to provide unique one-on-one support to help improve people's ability to cope with ALS and increase their sense of hope and community. IMALS also hosts virtual support groups each week. IMALS sees policy change and increasing federal funds for ALS research as incredibly important tools to finding treatments and cures for ALS, as well as improving the quality of life for those living with and impacted by ALS. Our annual legislative goals are to fully fund the Act for ALS, secure CDMRP funds, and pass relevant and impactful legislation. Now on to how IMALS aligns with the NINDS ALS Strategic Plan. There are 3 priorities within this plan where IMALS contributes. IMALS works toward increasing the biotherapeutic pipeline in 3 different ways. First, our clinical trials volunteer team meets regularly with drug sponsors at different points in the drug and clinical trial development to offer insights into designing a clinical trial that will enroll people quickly and avoid design components that will frustrate and deter participation or not provide the results the trial designers need. To set a standard and assist in conversations with drug sponsors, this team created the patient-centric clinical trial design PACTED rating criteria with 3 major areas: optimizing access to investigational therapies, advancing scientific progress, and the trial's patient friendliness. Currently, clinical trials team members use it to guide the creation of and evaluate existing clinical trial designs. They publish these evaluations online to help people living with ALS find the right clinical trial. Additionally, IMALS supports and is actively working on passing the Promising Pathway Act, as it would provide a way for promising biotherapeutics to get to the people who need them faster. IMALS works toward defining the natural history of ALS in 3 different ways. First, IMALS staff and volunteers educate the ALS community on genetic testing and discuss the social, cultural, and economic barriers to getting tested. Second, ALS Signal is a dashboard that convenes disparate information about clinical trials to help people living with ALS and their clinicians find the best possible clinical trial or expanded access program for them. Lastly, we empower and encourage ALS advocates to join patient and community advisory boards and grant selection and policymaking committees to ensure key stakeholders and decision makers have the information they need to make informed decisions and programs for the communities they serve. IMALS works toward optimizing the quality of life for people living with ALS and their caregivers in 4 different ways. Firstly, IMALS's peer support initiative, ALS support team, and support groups all contribute to the positive physical, social, and mental health of people living with ALS and their loved ones by providing numerous ways to receive support. One-on-one with a peer, one-on-one with a social worker, and within groups of peers and a social worker. The peer support initiative also contributes positively to mentors as it gives their struggles a purpose and meaning in the same way that the volunteers on IMALS's community teams improve their social and mental health by becoming connected with peers, social support networks, and providing them with a purpose and a higher meaning. All 3 of these are designed to address loneliness and isolation. Support groups also offer space for connecting and sourcing ideas for navigating daily life, so people don't need to recreate the wheel when it comes to functional living. IMALS looks forward to continuing to work on these and other priorities with the NINDS and ALS community. Thank you for your time, and please reach out to us at community@imals.org.
Hello, I'm happy to share a little history with you about Answer ALS and our resulting data platform, NeuroMind. Here is a brief summary. In 2013, the idea was proposed at the Tim Gleason Summit to create a business plan that would help bring an end to ALS as we know it today. That business plan is now Answer ALS, one of the largest and most comprehensive ALS studies. From this study, on January 8th, 2020, we launched this massive collection of ALS data and have made it freely available to researchers around the world through the unprecedented NeuroMind portal. As briefly stated before, here is more detail on the Answer ALS program as illustrated by this image. We've enrolled a total of 1,047 participants across 8 clinics. This includes both study participants and controls. To fortify our data set, we added an additional 228 controls. Every participant that came through the clinic underwent an initial evaluation, contributing blood samples for iPSC generation and whole genome sequencing. Many continued to be seen in the clinics every 3 months for follow-up assessment, making up the longitudinal data set. These iPSC lines were used to produce motor neuron lines, setting the stage for deeper omic analysis. A generous subset of participants also shared biofluids like CSF, plasma, and serum, all of which are banked and available through MGH. All of the acquired omic and clinical data has been made accessible via the NeuroMind portal, promoting open sharing and collaboration. The participant data then links to the cell line repository through the platform. Quickly highlighting our program's key developments over the approximately 1.5 years since NeuroMind began, NeuroMind has received over 50,000 visitors to date. We have over 550 data access requests. Available omic data stands at 95 terabytes with a projected total of 150 terabytes. Over 340 independent projects have been initiated using Answer ALS datasets, a number based solely on the formal applications. Data transfers are 800 terabytes of data that has been shared with global researchers. Of the 862 iPSC lines the team has completed, 789 are available for acquisition. Over 20 publications have cited or detailed the Answer ALS data. Now, despite NeuroMind's temporary inaccessibility in 2022, our commitment remains to provide researchers easy access to cell lines and their associated participant data. Our key takeaway, beyond the abundant resources we've generated, our proudest achievement is streamlining the access to cell lines and related biological materials, aligning with correspondent participant data. This slide represents a timeline of the last 10 years of a commitment to the process that was outlined. What started as a back-of-the-napkin idea by researchers, clinicians, and patients remains steadfast in its path and fulfillment of openly and freely sharing all data and samples to help accelerate this research. Over the course of building what was believed to be a $25 million program has ultimately resulted in $45 million effort once the true depth and study was considered, committed to, and realized. I won't spend much more time on this slide, but to emphasize how important it is to have the ability to have a strong financial team working in concert with the entire program. It's almost as if we have business leaders who have been living with ALS that are guiding us, and frankly and unfortunately we do. Also wanted to give you an introduction to a wonderful collaboration we have developed and are in the final phases of implementation, and this is with the Alzheimer's Disease Data Initiative team. Alzheimer's Disease Data Initiative, or ADDI, is a 503c medical organization focused on Alzheimer's research and is now expanding data sets to other neurological diseases. We partnered with ADDI to bring free cloud-based workspaces and tools to our own data users. We want to encourage collaboration and sharing of data derivatives and tools in the workbench to speed up discovery. This platform is open to a global community of neurodegeneration researchers and data contributors. The idea is ALS data users will be able to access the workbench, either through the NeuroMind portal or ADDI's. An approved ALS data user will be able to use the cohort selection tool to down select to those participants and data they are interested in analyzing, and then select to go to the workspace and process analyze the data for free. This project is being developed and moving to implementation this year. Putting all this together, we can get an idea of how the ALS Research Hub works. A typical user journey starts by engaging the ALS data portal to explore metadata and cohort attributes. This is where the user can select which participants and data they would like to interact with. The postmortem participants and data will be easy to locate with this tool. The user then has a choice to one or more of the following: download metadata or big data, order biosamples or cell lines by connecting with research results to sites such as Cedars-Sinai, analyze participant data in visualization tools such as the new heat map tool we are releasing. They can then refine their list based on results from analysis or ordering availability. Using this new list, they can return to the portal for more specific metadata. Additionally, a user can use the data and/or material gained from ALS in their research and validation efforts. New models and hypotheses can be gained and shared with the larger scientific community through open workspaces like ADDI's Workbench. Those models can then be used to go back to the data and search using a new set of attributes found through validation experiments. This becomes an ecosystem for data exploration and discovery. Taking the holistic view, it's easy to see how Research Hub for ALS might help with discovery. And finally, throughout our 10-year journey, we could not have been successful without our partners, funders, and study participants. We thank them and you for the continued commitment, and we thank you for this opportunity to share a little bit about ALS.
I'm Neil Thacker from the ALS Association. Thanks for the opportunity to speak with you today, and thank you to the scientists and the volunteers and the NINDS staff who put this ALS plan together. As you probably know, the ALS Association is dedicated to making ALS a livable disease. By that we mean a fundamental transformation in the experience of what it's like to live with ALS, a much longer lifespan, a much higher quality of life, and ways to prevent people from developing ALS. The association runs an advocacy program and a care services program, but I want to focus on our research program today. And at any given time, we're funding over 140 studies around the world. And we have a list of them at the link, but we do everything from basic science to preclinical work to clinical trials, everything but phase 3, which is out of scope for us, And then also managing ALS in the real world, ways to figure out really how to make ALS livable by trying to find research and interventions and improvements for people who are living with the disease today. Assistive technology, telehealth, real world data, and quality of care studies and ways to prevent the complications that come with ALS. As we've continued our work in making ALS livable and finding cures for everyone, we've come up with some general recommendations for the ALS research community. First, as you know, the new treatments in ALS has created more change and more improvement in ALS clinical care than ever. And so we have a real opportunity to figure out how to make all of these new drugs work as effectively as possible in the real world. What's the right combination of therapies for the right individuals, a particular form of ALS? We need to figure that out quickly. Second, we need an intervention pipeline for prevention, just like we have for developing drugs to treat people who already have ALS. We need to take everything we know about risk factors for ALS and genes that may cause ALS and come up with interventions. Again, we need to do this quickly. And finally, what NINDS can do, what NIH can do, is fund the workforce for clinical research that can help everyone with ALS in the United States get the best standard of care and participate in research. Right now, those opportunities aren't broad enough. And so I want to talk about that a little bit more on the next slide. And so I want to focus a little bit on this gap I mentioned before between what's the best possible care, all the approved treatments and multidisciplinary care, and the care that most Americans get. There is a gap here, and we don't know how to solve it, and so it's a great opportunity for science to step in and solve that problem. And just as the National Cancer Institute spends about a quarter of its budget trying to reduce this gap, I think NINDS has the same opportunity here to reduce this gap and help more people living with ALS get more effective treatment and better outcomes, even with the technology we have today. And so I want to come back to this question about how to get benefit out of a research portfolio. And to do so, you have to figure on not only what we're studying, but when we think that study is going to result in human benefit. And so if you want to focus your research investment on basic science, you have to recognize that there is a long time, maybe even decades, before that research is going to have an impact on human beings. And as you get closer and closer to the real world, you get into the preclinical space and even the clinical space, that time shrinks. And then when you get to care that's actually out in the real world already, that time is the shortest at all. And here, maybe 2 to 5 years if you're going to do research on quality of care or phase 4 data. And so, for example, if you're studying an issue like developing a way to reduce the time to diagnosis, you could start at the very basics and try and work out a biomarker or a diagnostic process all the way from the beginning, and that's going to take decades. If you want to work on a biomarker that's already been identified in theory and try to bring it into clinic, that could happen in a shorter amount of time. But if you really wanted to have an impact on time to diagnosis right away, you can figure out how to take people who might have ALS and need diagnostic services and get to them to the people who already know how to diagnose ALS and do it all the time. And that's a service research problem that has a very short time to benefit. And so ideally, what you would be thinking about for a problem like reducing time to diagnosis is doing all of these things, a balanced approach. And in your portfolio, you're consciously and deliberately balancing time to benefit along with all the other considerations you make when you fund research. And that's what's missing in our ALS strategy across our different funders, and I'd like to see more of that. So thank you for the time, and thank you for the opportunity to talk with you today.
You can just bear with us for one quick second here. It seems we're having some brief technical difficulties with this last video.
Thank you for this opportunity to describe how the Muscular Dystrophy Association is addressing different aspects of the NIH strategic plan for ALS. Although ALS is not in the name of the Muscular Dystrophy Association, it's been a major part of MDA's fight against neuromuscular disease since the 1950s when Eleanor Gehrig requested the help of the association to better understand the disease and develop therapies for it. In this respect, MDA research funding has led, in part, to almost every major inflection point in the field, such as the identification of SOD1 mutations as the first genetic cause of ALS, the development of the SOD1 mouse model, the development of Rilutec, the first approved drug for ALS, support for the first antisense oligonucleotide developed against SOD1, a precursor to Qalsody, identification of TDP43 as key to the pathology of ALS, the role of C9orf72 repeat expansions in ALS, the development of Radicava, the second approved treatment for ALS, the first trial of T regulatory cells in ALS, the Healy ALS platform trial, the MOVER ALS clinical database, and early testing for one of the components of Relyvrio. The NIH strategy for ALS addresses accelerating the biology behind this disease. In addition to the work described previously, the $174 million MDA has invested in ALS research includes $6.2 million in uncovering molecular mechanisms underlying ALS clinical heterogeneity, $2.1 million in identifying causes of sporadic ALS, $10.4 million in harnessing emerging technologies for enhancing knowledge of ALS disease mechanisms, such as machine learning approaches, longitudinal live cell imaging applications, and wearable devices, and an additional $12.6 million in other ALS biology-focused projects. Establishing a network of ALS Centers of Excellence was also identified as a strategic outcome. MDA funds a network of over 150 designated care centers that meet MDA's requirements for multidisciplinary care in neuromuscular disease. Of these, 48 are designated ALS Centers of Excellence, and over 10,000 ALS visits throughout the network were recorded in 2022. MDA's MOVER Clinical Data Hub is an observational research study that collects longitudinal clinical data. MOVER currently contains data from over 2,600 participants with ALS, representing over 9,600 individual encounters. MOVER data is HIPAA and Fair Data Principles compliant and has been used to support regulatory submissions. MDA also supports ALS data repositories at Emory University and at Columbia. MDA works actively with collaborators, including industry, to support new initiatives in ALS research. For example, we are currently collaborating with Mitsubishi Tanabe on the ALS Go Digital study, which will marry remote collected digital data with clinic collected ALS data from MOVER. The goal is to help efficiently validate remote outcomes in order to decrease geographic and other barriers to trial participation for those with ALS. MDA has contributed significant funding to support clinical trials in ALS, including the Healy ALS platform trial, pre-symptomatic ALS studies at the University of Miami, the development of new diagnostic tests for ALS at InVector, and the testing of a neurostimulatory device by Pathmaker Neurosystems. MDA has built robust programs to support people living with ALS, including educational and learning opportunities, such as the ALS Family Roundtable, at-home PT/OT guides, and community webinars. MDA also maintains numerous professional education opportunities on topics such as clinical research updates, ALS management, and ancillary care. MDA has an annual clinical and scientific conference, which features an ALS track and is attended by over 1,200 people in person and another 600 virtually. Anyone with a diagnosis of ALS or their friends and family who are registered with MDA can attend the virtual conference at no cost. MDA remains committed to improving the lives of those living with ALS and is looking forward to supporting the NIH in the execution of these strategic priorities. Thank you.
Dr. Gubitz.
Yeah, thank you for all the nonprofits who have sent in their really excellent presentation. It's just so amazing and encouraging to see all the summaries of the research programs. And when we sort of worked with all of you last year to develop strategic priorities, we always felt that because the recommendations, they're so broad, across different research domains that no funder can do this on their own. So we are always thinking of the strategic priorities for the field, where every organization brings in their expertise, their know-how, and then also their perspective and specific focus to help us advance the strategic priorities. And I think the presentation that we just saw are really a testament to that. And hopefully we'll still get the video from the Packard Center up. So that's a group that is very strong in basic research and funding a lot of really excellent academic research projects that we'll see, but otherwise maybe we'll send that video later to you after today's presentation. So we have been very busy answering many of the questions that we received through the Q&A. So thank you all for your engagement and sending in really excellent, important questions. I think we already answered 27. There are a few left. And so since we have a little bit of time here, maybe sort of I can just give an answer, for instance, to one of the questions about model systems and about NIH's interest in developing model systems for sporadic disease. So that's something that, you know, we take very seriously. I think a lot of knowledge in the field sort of has built on the discovery of disease genes, but we understand that the majority of patients do not carry one of the known causative mutations. And, you know, this is sort of a really important area of interest to understand sporadic disease. And there are new technologies that now allow us to address that. And that's sort of, you know, deriving these cell systems from patients who have sporadic disease. We have investment in multiple, you know, research grants to academic investigators, developing these cell systems and then understanding what, you know, cellular processes are affected. And then also there are sort of these organoid systems being developed where it's not only one cell type, but multiple cell types, and sometimes even sort of blood vessels to sort of mimic sort of a mini spinal cord, for instance, or a mini brain organoid. So there's a program that we, NINDS, collaborate with one of our partner institutes called NCAT to advance these technologies. And that can be done for familial forms of ALS, but definitely also sporadic disease. So certainly a really important area for NIH. It's an excellent question from one of our webinar participants. So keep on bringing in the questions and we'll sort of hope to answer them as they come in. I think, let me look through the Q&A if there is still something unanswered that we should respond right now. I mean, one question, is this working now at the Packard Center? And one question sort of I have in mind as
Professor of Neurology and Neuroscience at Johns Hopkins and the director and founder of the Robert Packard Center for ALS Research at Johns Hopkins. I've been asked to provide a brief overview of how we as a center and an organization contribute to ALS research over the years. We were established 25 years ago, and principally we are an academic consortium that focuses on developing understanding in preclinical model systems the pathogenesis of ALS and use those systems to develop and discover new therapies. The core focus over the years has been to bring leading researchers, both junior and senior, together to develop the tools that we need as researchers to understand what causes ALS and how we might develop future interventions. The vast majority of our participants are researchers from the US and Europe, not just at Johns Hopkins. And we're set up so that we meet monthly as researchers with a lab-like presentations and a very large academic meeting, probably the largest preclinical science meeting in ALS, bringing together 300 plus basic researchers from Europe, the United States, and elsewhere, as well as their trainees. The point of our research, of course, is to develop an understanding of ALS as well. And aside from that research team and the desire to share all of our tools, we also have a portfolio of tissues and preclinical models that are shared worldwide. The vast majority of ALS pathways since the discovery of the SOD1 gene have been discovered by academic researchers And largely through the tools, the model systems that we have made through the Packard Center, this slide highlights almost half of all mouse and preclinical models, IPS models in rodents that have been developed by Packard Center researchers, which is really our mission: develop those tools, understand those pathways, and develop drugs. To advance the understanding of ALS, especially as we came to understand that there might be truly subgroups of patients, biological along with our clinical, I founded and developed the Answer ALS Research Program, which is partnered with the Answer ALS Foundation to collect longitudinally the largest collection of ALS patients ever amassed, over 1,000 patients nationwide, follow them longitudinally, collect biofluids from all patients longitudinally, and importantly to develop, as shown on the slide, iPS cell lines from all patients, convert those cell lines to their spinal neurons, and as such act as a liquid biopsy, if you will, or a biopsy-like setting, where we analyze those tissue samples for proteome, RNA-seq, epigenome, as well as whole genome on patients, and as well as a collection of data from an app that we developed with IBM Watson. In aggregate, this amounts to 6 billion data points per patient. And with the help of Microsoft and the Answer ALS Foundation, all of the data, as well as IPS lines, have been freely made available to both academic and clinical researchers worldwide. As an example of that, and this is now a little out of date, we've now shared and generated almost all 1,000 IPS cell lines, generated epigenomics, proteomics, and transcriptomics from all of those lines as we go forward. There's more to be developed. And that data has now been shared really worldwide through 32 different countries, more than 200 projects, 250 projects started, as well as shared with close to now 100 companies worldwide to use both the platform of living cells as well as the data generated from those to understand ALS and move forward. There are no IP restrictions on the use of those cells or data. It's freely accessible. And today, more than 95 terabytes of data have been released to researchers around the world, with 800 terabytes downloaded to research labs in academics as well as companies. Importantly, that data is matched with IPS cell lines as well as a limited set of autopsies. So we can match these living biopsies, if you will, the IPS lines, to true patient autopsies. As an example, Answer ALS, the foundation, has provided a really great video that we promote to our researchers worldwide.
On January 10th, 2017, Jay Quinlan got up at 5 a.m., did his breathing exercises, stretched his entire body, took 20 minutes to dress himself, brushed teeth, flew 3 hours on a plane, got into a van, drove to a clinic to be stuck with needles, and got bandaged up before flying his sore and aching body hours back home to collapse. Why? So Jay could donate 6 billion data points to Answer ALS in order for you to research them. When it comes to ending ALS, Jay's done his part. And with the NeuroMind data portal containing over 2 trillion data points, we're ready to help you do yours. Visit answerals.org and register to do your part to end ALS. On Jay.
So from some of that data, we already have a number of clues about whether it's subsets or druggable targets. One of the discoveries made by a group at Hopkins and with collaborators is the obvious defect in nuclear pores that have been well worked out in both sporadic and C9 ALS. And a further understanding of that pore defect has expanded to other diseases. We'll focus really briefly on ALS, but other related but different defects have been found in FTD and AD as well as Huntington's disease. So it's a platform that's gone beyond ALS. Importantly, we know that all ALS patients, sporadic, have a defect in TDP43. It's nuclear, becomes cytoplasmic, and then eventually aggregates. Although the field initially studied the aggregates, we now know that early in the disease, it's a nuclear clearing, and that defect looks to be a defect in nuclear pores, which have been discovered really through the biology of studying nuclear pores in sporadic and familial ALS cell lines. We've come to understand that defect. It's a defect in a pathway that creates damage to the nuclear pores, involved a pathway known as escort and CHIMP7. And with those IPS cell lines, you're looking at a profile here of now 157 different patient IPS lines that we've been able to study. And these different colored columns reflect all the different defects in TDP43. And as one can see very quickly, is every patient's a little different. They're not uniform, although they all have a defect in nuclear pores and CHIM7 and variable defects in TDP43. That defect is actually matched in human brain tissue because the Answer ALS collection has matched human tissue. By the way, that tissue is freely available to researchers. And interestingly enough, that platform teaches us that IPS cells beautifully match actual brain tissue from autopsy. Is the least from this pathway, teaching us that the IPS platform is a phenomenal platform to understand pathway defects in ALS. And actually using that platform, we've been able to test drugs, we the community, and drugs that fix the nuclear pore in a almost like clinical trial of 30 different patients, C9 and sporadic. That pathway, when mitigated with an ASO, completely repairs the TDP43 defect. And therefore, the IPS platform from the Packard Center, from the Answer ALS collection, has been already used to identify patients and a druggable pathway that looks quite intriguing in patients. Lastly, in plans for an expansion of the Answer ALS and the Packard Center, we're going to build an autopsy platform. We know that there are existing autopsy platforms that are relatively small, but we now have even a larger platform planned where we've now collected over 1,500 autopsies with matched IPS cell lines, matched biofluids. All of this will become freely available eventually with, of course, proper funding from external sources for this program, just like the Answer ALS platform was funded through an Answer ALS effort in the Packard Center. Lastly, new data sets are coming to the Packard Center and the Answer ALS platform, including an enormous proteome screen. All of the Answer ALS samples, over 1,000 participants that include longitudinal samples, have now undergone proteome. That will be freely available and it'll be matched with 30,000 AD patient data sets as well. NFL from this largest collection longitudinally of ALS patients has now been completed, serum NFL. That again will be freely available, and soon we'll have metabolomics. So in a nutshell, the Packard Center really provides both the tools and the data sets longitudinal on a very large scale for the ALS community. Thank you.
All right, so we are just about out of time. I would like to give the floor back to Dr. Walter Koroshetz for any final comments. I would like to mention that we have learned that the Muscular Dystrophy Association video also had some technical difficulties. We will send that MP4 file out to everyone who registered and participated in this webinar. We apologize for those technical difficulties. Dr. Koroshetz.
Oh, yes. So, gee, apologize to the MDA. Yeah, we'll definitely get that video out. Sorry about that. No, just to close, I think if you weren't convinced that ALS community is really geared up and moving the science forward, this was the time to change your mind. It's I think it's just so heartening to hear what the different groups are doing. And here at NINDS, we're blessed to be able to work with you. And our plan is to, to kind of raise all boats. And the patient is really at the center of what we're trying to do. We'll use every lever we can to improve the care, quality of life of people with ALS, and to find treatments that just stop this disease from affecting people as it does. So I want to just thank everyone for the time you put in, and we're going to be working together hand in hand over until we get a cure for this disease. So thanks very much.